Hi, I'm Emerald Robinson. In this What Is video, we're going to take a closer look at atoms. All matter is made of elements, substances that cannot be broken down by chemical activity. An atom is the smallest particle of an element that retains the element's properties. Atoms are the building blocks for everything in the universe, including ourselves. People often think of atoms as the smallest possible forms of matter, but even smaller particles make up atoms. Each atom has a dense center called a nucleus. The nucleus contains neutrons, which have a neutral electric charge, and protons, which are positively charged. The number of protons in an atom determines an element's physical properties. No two elements have the same number of protons. Revolving around the nucleus like planets around the sun are negatively charged particles called electrons. Electrons and protons attract each other, so atoms usually have equal numbers of the two particles. Scientists assign atoms an atomic number and atomic mass unit. The atomic number is the number of protons present in the atom. For instance, a helium atom has 2 protons, so helium's atomic number is 2. Add the number of protons and neutrons in an atom's nucleus together, and you get the atomic mass unit, or AMU. A helium atom has 2 protons and 2 neutrons, so has a mass of 4 AMU. Electrons have almost no mass and are not included in AMU calculations. While an atom's number of protons remains constant, individual atoms can lose or gain electrons. Atoms with different ratios of electrons to protons are called ions. Atoms of the same element can also have extra neutrons. Scientists call atoms with extra neutrons isotopes. Despite having different neutron counts, isotopes retain their element's chemical properties because the number of protons remains constant.
What are enzymes? Enzymes are catalysts. They make things happen and speed up the action, but without being changed themselves in the process. A catalyst is a chemical involved in, but not changed by, a chemical reaction. Enzymes are proteins that perform a special function. They act as an organically grown catalyst to launch a chemical reaction that either forms new chemical bonds or breaks down old ones between different molecules. Enzymes do this by reducing and weakening the energy barrier that normally exists between atoms, a barrier that otherwise prevents or delays these atoms from forming a bond with each other. Thus, without enzymes, necessary reactions in our body would occur too slowly or not at all. The amino acid sequences primarily determine how the protein folds to its 3-dimensional shape, which is critically important for its catalytic function.
We're going to be talking about elements, atoms, really why it's all about the electrons, and some Lewis dot structures, which, in my opinion, may be the key to happiness. When we get started here, we're really talking about what stuff is made of, and stuff is made of matter. From here, you can really kind of go off a cliff in terms of how far you want to go into this. This is really the realm of physics. So I just want to say from the outset that this is a highly abbreviated version of an understanding of what the stuff is in the universe. Matter is made of elements. Elements are pure chemical substances that you can't break down into anything else. Now, we organize elements in this amazing table called the periodic table of the elements. It's really amazingly organized, and I'm just going to say it. It's beautiful. Okay, how much of a geek am I to say that? But it really is, because if you understand how this table is organized, this is tremendously powerful. Notice I didn't say if you memorize it. I do not have my students memorize any part of this table. That's what it's there for. That's why you can look it up. But you have to understand how to use it and what it means. In terms of elements in the universe, the vast majority of them are the 2 smallest elements, hydrogen and helium. On our planet, in the Earth's crust by weight, we've got oxygen, silicon, and aluminum. If you take the Earth as a whole, not just the crust, by weight, the most abundant element is iron. In terms of biology, only 25 elements are essential for life. And of those 25, only 4 make up the vast majority of most living things, including penguins. So if you're wondering, well, what are we made of? You'll be thrilled to know that you can actually buy a shirt with human ingredients on it. Oh my gosh, I love these geeky gifts. I don't know what that barcode is at the bottom. But anyway, so here's a nicer view, table of the elements of the human body. And again, you can see that over 96% of humans are made of oxygen, carbon, hydrogen, and nitrogen. So if you go back to the periodic table, and it is kind of overwhelming, I admit, but you remove all of the elements that are not found in humans, it's really not too bad at all. This looks somewhat manageable, right? Understanding the periodic table also allows you to understand all these stupid geeky gifts, like this bacon shirt, which I don't know, I was just in a weird mood. Okay, so the smallest unit of an element, which retains all of the properties of that element, is called an atom. That word comes from the Greek, a term which means not to be divisible, meaning you can't cut it up into smaller pieces and still retain the properties of the element. We can actually see atoms today. This is a really cool transmission electron micrograph done by the physicists at UC Berkeley. The red arrows are hydrogen. That black arrow is a carbon atom shown on the surface of graphene. Now, in your textbook, you will see atoms drawn like this. Electron clouds, the shell or Bohr model. And I got to be honest with you, this is just not true. Atoms do not look like this. We use these drawings because they represent sort of an easier way for us to talk about them, but this is not what atoms really look like. Atoms are really hard for us to wrap our brains around because they are unbelievably small. That black bar right here at the bottom represents 1 angstrom, or 100,000 femtometers. So an angstrom is 10 to the minus 10th meter. And the vast majority of the atom is actually empty space. So over 99% of the mass of the atom is concentrated at the center in the nucleus. And the rest of it, where the electrons are, the orbitals, are just hugely spread out. And it is really difficult for us to really draw and think about what atoms really look like. So I have a couple of analogies for you, of course. One is an analogy of scale, and this is a soccer field. And if you take the nucleus to be the size of the soccer ball, then an electron could be a fly buzzing around the spectators. And most of the stadium represents the space between the atomic nucleus and the electron. So most of the atom is empty space. Now, this analogy is not really accurate either because it shows the electron as a particle, as a fly. And actually, that's not true. One of the best analogies that a chemist gave me for this is with cotton candy. And he said, okay, where's the sugar? Well, the sugar's kind of everywhere. I mean, the whole cotton candy is made of sugar. That's kind of the same idea with these electrons. So the orbital itself really is the electron, but that's really hard to draw and describe. So what I always say is, strive for understanding, strive for the real truth, but we, like all of your textbooks and basically everybody else, we're going to use these models and drawings. We're going to draw the little electron as like a ball or a bead so that we can keep track of everything. Okay, but realize that that's not really the truth. So getting back to the atoms and the periodic table, there's a couple of numbers that we need to concern ourselves with. One is the atomic number, and that's just the number of protons that an element has. And I'm picking carbon here as an example. The atomic number for carbon is 6. What that means is that carbon has 6 protons. That number does not change. It's kind of like your calling card. So if you add a proton to carbon, it's not carbon anymore. So now your atomic number would be 7, and now you're nitrogen. So the atomic number does not change. The mass number, or what we call the atomic weight, that's the number of protons plus the number of neutrons. The first thing that you notice is that these numbers are decimals. So maybe you're wondering, well, are there fractional protons and fractional neutrons? And, what's up with that? Well, what we're really getting at here is the mass number is actually an average. So the number of protons does not change, but the number of neutrons can change. So if we look at carbon in the universe, there are different numbers of neutrons in various carbon atoms that you could find. We call these isotopes. Most carbons in the universe have 6 neutrons. And of course, the number of protons doesn't change. So 6 plus 6 is 12. So the vast majority of carbons that you would find have the mass number of 12. You will also find, though, some carbon atoms with an extra neutron. And if you add 6 plus 7, that gives you the mass number of 13. Some atoms have 2 extra neutrons, 2 more than the normal number. And the mass number here is 14. This is probably the one that you're most familiar with. Carbon-14 is what we use for dating. Not dating, will you go out with me, but dating, how old is that tree? We use isotopes for all kinds of purposes, actually, in medicine as well as radioactive dating, because they decay at a constant rate. So if you look at the decay rate of carbon-14, this is what it means. So we have times 0, some amount of carbon-14 that's present. After one half-life, which for carbon-14 is about 5,000 years, after one half-life, you have half that amount still remaining. After 2 half-lives, you have half of that half. And after 3 half-lives, you got another half. So it's not like after 2 half-lives, it's all gone. That's a big misconception. So what you should notice is this exponential rate of decay. And this rate is constant. So that's why we can use isotopes to date living tissue, to date rocks, and things like that. But enough about the nucleus. Chemistry is really all about electrons. So we're going to spend the rest of this video talking about them. Now, atoms like to be stable. And in order to become stable, sometimes they have to play with other atoms. In this way, they're kind of like people. But you might ask the question, does everybody need to play with others in order to be happy? Humans are social creatures, so most of us do, but there are actually individuals that don't. And the same thing is true in the periodic table. We have to the far right the noble gases. Noble gases, so named because, like nobility, they do not mix with the commoners. And in this video, we're going to explore why. Why don't they form bonds with other atoms? Everybody else on the table, they form bonds, but not the noble gases. You got to understand the electrons in order to understand why. So, electrons fill around the nucleus from the inside out. And what I mean by that is as you look at different atoms, and different atoms have different numbers of electrons, they will fill around those nuclei from the inside out, just like water fills in a glass. When you fill a glass with water, it fills from the bottom up. Okay, you can't do it any other way. If you can, I'd like to see that. Shell number 1 is going to hold 2 electrons. Shells 2 and 3 are going to hold 8. Yes, for you chemists, I am oversimplifying this. When you go on and take more general chemistry, you will see that the electron configurations are a little more complicated than that, but for the purposes of general biology, what I'm giving you should get you through most of the problems that you're going to come in contact with. I just got to say, don't get your heart rates up when you see stuff like this. Even at first glance, you notice that there is a pattern. So this is manageable. But we're not going to go into that in this video. We're going to focus on happiness. What you got to remember is that atoms are happy when their outer shell is full of electrons. The outer shell turns out to be so important, we actually give it a name. We call it the valence shell. So we're going to talk about how they get to be happy right now. As we go through this, we're going to see that carbon is not happy by itself, whereas neon is. We're going to see why. My analogy for this is a stadium in the round. So a stadium with a stage inside and spectators all the way around. And of course, we're going to have penguins in there too. So we're going to have a rule and a goal. The rule is we're going to sell tickets closest to the stage first. The goal is we want to make each shell around the stage full of spectators. Now, in our little theater, it's going to be a very little theater. The first shell is only going to have 2 seats in it. The second shell is going to have 8 seats, and the third shell is also going to have 8 seats. So just go with me on this, okay? It's a small, intimate theater. So here are our seats. We're going to look at different scenarios and see what it looks like. So here's the first scenario. We sell 1 ticket. So we're going to satisfy the rule. The tickets that we sell are going to be closest to the stage. The goal is not met, though, so that's not so happy. We have one empty seat in the first ring, and this is actually hydrogen. Another scenario, we sell 2 tickets. So the rule is they're going to be the ones closest to the stage, so that's all good, and we actually met the goal. The shell is full. So that's a really happy, stable scenario, and this is actually the element helium. What if we sell 7 tickets? So notice the rule, we're selling the tickets closest to the stage first, but you notice now that we have 3 empty seats in that second shell. So this is nitrogen. This explains why nitrogen would want to form bonds to try and fill those empty seats. What if we sell 10 tickets, though? Now you notice that the first 2 shells are full. This is a very stable configuration, and this is actually the noble gas neon. What about 13 tickets? So now we're moving into that third shell. We're always filling from the inside out, but now that third shell has only 3 occupied seats and 5 empty ones. So that's not so happy, and that is the element aluminum. In our last example, we have 18 seats, and you notice we have all 3 shells full. This is a very stable configuration. This is the noble gas argon. So maybe you've noticed that it seems to be about the valence shell. If the valence shell is so important, why don't we just focus on that? I mean, who cares what the core of the beast looks like? Let's just look at the outer shell, since that's what's going to determine how happy an atom is. If you're thinking that, that's awesome. You're not alone, because some other really smart people have thought exactly the same thing, like Dr. Gilbert Lewis, who is a physical chemist. He spent most of his career at UC Berkeley. And he did exactly that. He said we should draw an elemental symbol, and then we're going to draw the valence shell electrons around it. And I always tell my students, if you keep track of the valence electrons, you're going to be happy. So let's see how this works. We're going to look at carbon. The most important part in terms of carbon's reactivity is the valence shell. So let's just get rid of everything else. And we're going to draw only the electrons in the valence shell around carbon. That's the most important part. So carbon has the atomic number 6, and now you know that 2 of those electrons are in the first shell, leaving 4 for the valence shell. Now I just want to remind you those penguins represented electrons, but for heaven's sakes, don't draw penguins in your Lewis dot structures. You're going to get kicked out of class. So what we do is we draw literally dots around the element to represent the valence electrons, and ta-da! That is the Lewis dot structure for carbon. How easy is that? So let's look at nitrogen. So to draw the Lewis dot structure for nitrogen, all you do is you draw the symbol for nitrogen, and then you go around it and draw the valence electrons, starting at the top. Now how you draw these varies. This is the way that I was taught. Starting at the top and going around, nitrogen looks like that. So let's look at aluminum. So here's the symbol for aluminum. Aluminum has 3 electrons in its valence shell, so you draw them around going 1, 2, 3. That's aluminum. So finally, let's do argon. Argon has a full valence shell, so we're going to draw 1, 2, 3, 4, 5, 6, 7, 8. A full valence shell for argon, and it's as simple as that. So check this out. If you look down the column of all the noble gases and you draw the Lewis dot structures for each one, you will find that they all look the same because all of them have full valence shells. And this explains why all the noble gases are happy as they are, and they don't form bonds with other atoms. Because if the goal is to have a full valence shell and you already have one, you don't need to play with others. The only thing that might confuse you is helium, and what you have to remember is that helium is very small, atomic number 2, so it has only 2 electrons. And so you have to remember that that first shell is in fact full with 2, whereas everybody else going down this group, they will all have an outer shell full with 8. So let's practice. When you practice drawing the Lewis structures, all you need, without the penguins, is a periodic table. So lithium, atomic number 3, is going to have 2 electrons in the first shell, leaving you 1 in the second shell. So when you draw the Lewis dot structure, that's it. 1 electron in the valence shell. For oxygen, you've got 2 electrons in the first shell, 6 in the second, for a total of 8. So you draw your oxygen with 1, 2, 3, 4, 5, 6 electrons in the valence shell. That's your Lewis dot structure for oxygen. Sodium has 2 in the first shell, 8 in the second, 1 in the third, for a total of 11. Sodium is drawn just like that. So it's really not too bad. And even though most biology instructors don't really show you Lewis dot structures, you're going to have to do it in chemistry anyway. And truthfully, if you understand Lewis dot structures, everything else is going to make a lot more sense. So here are the first 20 elements and the Lewis dot structures for each. And what you notice is that as you go down the vertical columns, which we call groups, they all have the same Lewis dot structures. And that explains why they have the same chemical reactivity and why they'll form the same kind of bond. Isn't that cool? How powerful is that?
Whenever a substance is oxidized, it loses electrons. Any substance that is reduced gains electrons. In a reaction, let's say if you were to write a half reaction, the electrons are always on the right side of a half reaction for oxidation. For reduction, the electrons are on the left side. Now, let's consider the oxidation numbers. The oxidation number for pure elemental metallic sodium, for any pure element, is always 0. The sodium ion has a +1 charge, so its oxidation number is 1. Anytime the oxidation number increases, the substance is oxidized. If you look at the example on the right side, the copper has a +2 charge, so its oxidation state is +2. And then as it becomes elemental copper, pure copper metal, its oxidation state is back down to 0. Because the oxidation number decreased, it was reduced. In other words, it's reduction. By the way, any substance that is oxidized is also known as the reducing agent. Any substance that is reduced is also known as the oxidizing agent.
Glucose. Well, glucose is a molecule, and it's known as a monosaccharide, which is the most basic unit of a carbohydrate. It's the key source of energy for all cells and organs in our bodies. Foods high in glucose include fruits, veggies, and grains. Desserts and candy contain a lot of glucose as well. Many sweets have a lot of added sugar in them, and sugar is made up of a glucose and fructose molecule. As mentioned before, glucose is a major source of energy in our bodies. Glucose is absorbed into the bloodstream rather quickly, so it can provide quick energy to cells. Extra glucose can then get stored as glycogen in the liver or muscles, providing an energy reserve. As with most substances in the body, too much glucose can cause problems. Overconsuming any type of food, including foods with high glucose content, can lead to obesity, which in turn can lead to an increased risk of many diseases such as diabetes, cardiovascular disease, high blood pressure, and even respiratory problems. Once again, always about moderation.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
We're going to look at the big picture here. Overall, the equation, you may or may not have seen it already, looks like this. Most people think about breathing when they see cellular respiration, and it's actually related. It is because of what the cells are doing that you actually need to take in oxygen and why you produce carbon dioxide. So it is related to the respiratory system. I'm going to make a lot of analogies with money because people are familiar with finances a lot more than cellular respiration. Show me the money. Oh, this is my favorite currency in the whole world. Isn't it gorgeous? That's the yellow-eyed penguin from New Zealand. Now, your options for getting money, you can either make it yourself, be the boss, or you can get it from someone else and be an employee. Okay, you're going to use this money, of course, to buy goods and services, or you can save it for the future. Now, of course, when we talk about cells, we're not talking about money, we're talking about glucose. Glucose is what cells need in order to do work. You have a couple of options. Some organisms make glucose all by themselves, and we call those organisms autotrophs. Auto means self. Now, there are a couple different ways that you can make your own carbohydrates. You can use energy from the sun. We call those organisms photoautotrophs. And some organisms use the energy from chemical bonds, which they break, in order to make carbohydrates. The other option, if you can't make it yourself, you got to get it from someone else. And those organisms we refer to as heterotrophs. Hetero just means other. Either way, you're going to use the glucose to do cell work, or you're going to store the glucose for the future. Now, what we use the glucose for is shown here. About 70% is just to stay alive. Okay, so here's the breakdown by organ. A lot of people are surprised that the liver has a bigger energy demand than the brain does. We got physical activity, and then the digestion of the food itself also requires glucose. Now, here's the thing. The use of glucose doesn't directly fuel cell processes. You need to convert it into something else first. So you take the glucose, you run it through a series of reactions, and you convert it into ATP, and that's what cellular respiration is. My financial analogy, we have a penguin, of course, who works hard all day and wants to have some ice cream after work. He gets a paycheck. Now, he takes the paycheck and goes to the ice cream truck. But you know what? The ice cream truck won't take a paycheck. They want cash, right? You can't get ice cream with a paycheck. So what the penguin has to do is, of course, go to the bank. And the bank does a conversion and gives the penguin cash money in exchange for the paycheck. And now you can take your cash and buy ice cream, right? This is not a complicated idea yet, right? So the analogy here is that the paycheck is the food that you eat, the fuel molecules that we take in. We cannot use the energy from the food directly. They have to be processed. All those nutrients get broken down and then converted mainly by organelles in the cells called mitochondria, which we're going to spend time looking at today. And then we're going to convert that energy from the food molecules into ATP. How good are we at doing this? Well, if you take your paycheck and you go to a bank and you have the bank do the conversion, the bank should give you the exact same amount, right, as your paycheck. Unless you go to one of those, check cashing places where they charge fees and all that. You don't want to go there. The food that you eat gets converted, and if you had $100 worth of food, the amount of usable energy that you're going to get out of it in the form of ATP is only about $40. So we're about 40% efficient at breaking down the energy from the food that we eat and converting it into ATP. Well, you might be wondering what happens to the rest of it, right? So you got about $60 left. That energy is lost as heat in the conversion process. When you convert energy, you produce heat as a waste product. Here's the big picture. In our cells, we extract energy from carbohydrates through a series of reactions, beginning with glycolysis. Glycolysis occurs in the cell cytoplasm, and that is going to involve 9 steps, which will break glucose, a 6-carbon sugar, into 2 molecules of pyruvate, or pyruvic acid, which are 3 carbons each. Those molecules of pyruvate will go into the mitochondria. And that's where we're going to spend most of our time. Mitochondria are like the energy transformers of the cell. So we're going to see, in a big picture sense, how they extract energy from carbohydrates. The mitochondrion has 2 membranes, an outer membrane and an inner membrane, which is thrown into folds in order to increase surface area. The space inside the inner membrane is called the matrix, the mitochondrial matrix, and that's where the citric acid cycle is going to take place. There is also the space between the inner and outer membranes, and we call that space the intermembrane space, not too surprisingly. Now, the matrix, like I said, is where the citric acid cycle, it's also called the tricarboxylic acid cycle, or TCA, that's where this beautiful piece of work is going to take place. Oh, geez, don't worry. Don't worry, I'm just going to show you the highlights of this cycle. We're not going to look at any of the details. First thing, it's a cycle, okay? It goes round and round. The second thing is that pyruvate gets groomed. It gets chemically altered into something called acetyl-CoA before it enters the cycle. So I point this out because a lot of students, when they look at, the order of events, glycolysis, and then the citric acid cycle, and then the electron transport chain, and they say, well, what happened to pyruvate? Because pyruvate doesn't actually enter the cycle. It gets groomed, it gets chemically altered, and it turns into this thing called acetyl coenzyme A. So that's what happened to it. The third thing to notice is that bonds are being broken from these carbohydrates, and the carbons which are released combine with oxygen to form carbon dioxide. And this is the source of the CO2 that you exhale. Finally, most of the high-energy bonds from what was originally glucose but became pyruvate and then acetyl coenzyme A as it enters the cycle, most of those high-energy bonds get oxidized, okay? And if you remember, if something gets oxidized, somebody else got to get reduced. I got a redox video. I'll put that link in the description box below. So who's getting reduced are these electron carriers. So every time you see, for example, NAD plus going to NADH, that is the addition of electrons to NAD, reducing it to NADH. So what's happening is that the energy from the original carbohydrate is being transferred to these electron carriers, and they are going to leave the citric acid cycle, and they're going to interact with some proteins on the inner mitochondrial membrane in order to generate ATP. So here we are again in the mitochondria, and you see where the citric acid cycle or the tricarboxylic acid cycle is occurring, and pyruvate being fed in. I am not showing you all the inputs and outputs, okay? And now we're going to see where the electrons that are being carried by the electron carriers, where they go. And where they go is a series of proteins built into the inner mitochondrial membrane called the electron transport chain. Okay, so we've got NADH, FADH2, the NADs and FADs, right? They're carrying the electrons that they picked up in the citric acid cycle, and they're going to take them to the proteins in the electron transport chain up here. So the source of the electrons will be the electron carriers. And as the electron carriers give up their electrons, the electron carriers get oxidized and the proteins in the chain get reduced. So this first protein will pick up the electrons and then transfer it to this guy, and then the electrons transfer here, and they kind of play a game of hot potato, as it were. And as the electrons move through these different proteins, a little bit of energy is given off each time, and that energy is used to do work, and that work is to pump protons into the intermembrane space. That's what the energy gets used for. So, okay, all of these proteins have names. Some of them are sort of stationary. Some actually are mobile. They move around. But the bottom line is what these things do is they have increasing electron affinity. So they want the electrons more than the protein just upstream. So the electrons are moving through these regions, and a little bit of energy gets given off at various points, and that energy is used to create a proton gradient. So this intermembrane space gets packed with protons. And where the electrons finally end up, the final electron acceptor after the electrons pass through all these proteins, is oxygen. And that's why oxygen is needed for aerobic respiration, because that's where the electrons end up. And when electrons are added to oxygen, you are going to make water. It's called metabolic water production. So I'm going to clean this up a little bit and show you the proton gradient in the intermembrane space. This is, again, created by the electron transport chain. Notice the direction of the gradient. That is where the protons want to go. However, that intermembrane is not permeable to protons, so they cannot pass through it. They are unhappy. And this disequilibrium is the source of the energy that we're going to use to make ATP. The analogy is a hydroelectric power plant. So this is going to be analogous to how the electron transport chain pumps protons into the intermembrane space. These protons are going to find that there is a magic space that they can fit through. They're going to flow down their gradient in the direction they want to go, just like water flows down through the turbine because of gravity. And instead of a turbine that converts that movement into electricity, we're going to have a protein called the ATP synthase, which is going to convert the energy from the protons going in the direction that they want to go into ATP. Let's go back and check this out. So here is the ATP synthase, and in the center there is a core where the protons fit. And as the protons flow in the direction of the gradient, now they're going in the direction they want to go, the cell harnesses that energy and uses it to phosphorylate ADP to make ATP. How efficient is it? Well, if you have oxygen present and you run aerobic respiration, if you break down the bonds in one molecule of glucose, you can generate up to 32, some textbooks will say 34 molecules of ATP. Compare that to anaerobic respiration. If there is no oxygen present, you stick that same glucose into the mix, and what you get? Only 2 ATPs. So you can see how much more efficient the aerobic respiration pathway is compared to the anaerobic respiration. So aerobic respiration is like 16 times more efficient at stripping the energy from carbohydrates and converting that energy into usable ATP. You might imagine how back in the day, anaerobes were outcompeted by organisms that could run this new, more efficient method of extracting energy. Eukaryotic cells got much bigger, we evolved into multicellular organisms, and it just went up from there. So the overall view of cellular respiration, the inputs, glucose and oxygen. We run glycolysis, where we split glucose into 2 molecules of pyruvate. You get a little bit of ATP out of that, by the way. You get 2 molecules of ATP just from that. And if you don't have any oxygen, that's all you get. If you do have oxygen, you can run the citric acid cycle. And again, the big deal about the citric acid cycle is this is where most of the high energy bonds from what was once glucose get broken. And where the energy goes is to reduce electron carriers. Now, where the electron carriers take their electrons is the electron transport chain. And so most of the ATP produced in aerobic respiration comes from chemiosmosis, the production of ATP using that gradient. What comes out, where the atoms end up, is carbon dioxide and water, and the energy from what was glucose a long, long time ago gets converted into ATP.
In my humble opinion, the single most important biochemical reaction, especially to us, is cellular respiration. And the reason why I feel so strongly about that is because this is how we derive energy from what we eat or from our fuel, or if we want to be specific, from glucose. At the end of the day, most of what we eat, or at least carbohydrates, end up as glucose. In the future videos, I'll talk about how we derive energy from fats or proteins. But cellular respiration, let's go from glucose to energy and some other byproducts. And to be a little bit more specific about it, let me write the chemical reaction right here. So the chemical formula for glucose, you're going to have 6 carbons, 12 hydrogens, and 6 oxygens. So that's your glucose right there. So if you had 1 mole of glucose, let me write that. That's your glucose right there. And then to that 1 mole of glucose, if you had 6 moles of molecular oxygen running around the cell, then, and this is kind of a gross simplification for cellular respiration. I think you're going to appreciate over the course of the next few videos that one can get as involved into this mechanism as possible. I think it's nice to get the big picture. But if you give me some glucose, if you have 1 mole of glucose and 6 moles of oxygen, through the process of cellular respiration, and so I'm just writing it as kind of a big black box right now. Let me pick a nice color. So this is cellular respiration, which we'll see is quite involved, but I guess anything can be if you want to be particular enough about it. Through cellular respiration, we're going to produce 6 moles of carbon dioxide, 6 moles of water, and this is the key, this is the super important part, and we're going to produce energy. We're going to produce energy, and this is the energy that we're going to that can be used to do useful work, to heat our bodies, to provide electrical impulses in our brains, whatever energy, especially a human body needs, but it's not just humans. It's provided by this cellular respiration mechanism. And when you say energy, you might say, "Hey, Sal, in the last video, didn't you just?" Well, if that was the last video you watched, you probably saw that I said that ATP is the energy currency. For biological systems. And so you might say, hey, well, it looks like glucose is the energy currency for biological systems. And to some degree, both answers would be correct. But to just see how it kind of fits together is that the process of cellular respiration, it does produce energy directly, but that energy is used to produce ATP. So if I were to break down this energy portion of cellular respiration right there, some of it would just be heat, it just warms up the cell, and then some of it is used, and this is what the textbooks will tell you. The textbooks will say it produces 38 ATPs that can be more readily used by cells to contract muscles or generate nerve impulses or do whatever else, or grow or divide or whatever else the cell might need. So really, cellular respiration, to say it produces energy, a little disingenuous. It's really the process of taking glucose and producing ATPs with maybe heat as a byproduct, but it's probably nice to have that heat around. We need to be reasonably warm in order for our cells to operate correctly. So the whole point is really to go from 1 mole of glucose, and the textbooks will tell you, to 38 ATPs. And the reality is this is in kind of the ideal circumstances that you'll produce 38 ATPs. I was reading up about it a little bit before doing this video. And the reality is, depending on the efficiency of the cell in performing cellular respiration, it'll probably be more on the order of 29 to 30 ATPs, but there's a huge variation here, and people are really still studying this idea. But this is all cellular respiration is. In the next few videos, we're going to break it down into its kind of constituent parts, and I'm going to introduce them to you right now, just so you kind of realize that these are parts of cellular respiration. The first stage is called glycolysis. Glycolysis, which literally means breaking up glucose. And just so you know, this part, the gly for glucose, or the glyco for glucose, really. The glyco for glucose. And then lysis means to break up. When you saw hydrolysis, it means using water to break up a molecule. Glycolysis means we're going to be breaking up glucose. And in case you care about things like word origins, glucose comes from the gluc part of glucose comes for Greek for sweet. And glucose is indeed sweet. And then all sugars, we put this ose ending. So that just means sugar. So you might think it's kind of a redundant statement to say sweet sugar, but there are some sugars that aren't sweet. For example, lactose. Milk, it might be a little bit, but when you actually digest lactose, then you can turn it into an actual sweet sugar, but it doesn't taste sweet like glucose or fructose or sucrose would taste. But anyway, an aside. But the first step of cellular respiration is glycolysis, breaking up of glucose. And what it does is it breaks up the glucose from a six carbon molecule. So it literally takes it from a 6-carbon molecule. Let me draw it like this. A 6-carbon molecule that looks like this, and it's actually a cycle. Let me show you what glucose actually looks like. This is glucose right here, and notice you have 1, 2, 3, 4, 5, 6 carbons. I got this off of Wikipedia. Just look up glucose and you can see this diagram if you want to kind of see the details where you can see I have 6 carbons, 6 oxygens. That's 1, 2, 3, 4, 5, 6, and then all of these little small blue things are my hydrogen. So that's what glucose actually looks like. But the process of glycolysis, you're essentially just taking I'm writing it out as a kind of a string, but you could imagine it as a chain, and it has oxygens and hydrogens added to it, to each of these carbons. But it has a carbon backbone, and it breaks that carbon backbone into two. That's what glycolysis does right there. So you've kind of lysed the glucose and each of these things. And I haven't drawn all the other stuff that's added onto that. These things are all bonded with other things, with oxygens and hydrogens and whatever. But each of these 3-carbon backbone molecules are called pyruvate. We'll go into a lot more detail on that. But by glycolysis, it by itself generates, it needs 2 ATPs. So it needs 2 ATPs, and it generates 4 ATPs. So net, on a net basis, it generates 2. Let me write this in a different color. It generates 2 net ATPs. So that's the first stage. And this can occur completely in the absence of oxygen. I'll do a whole video on glycolysis in the future. Then these byproducts, they get re-engineered a little bit, and then they enter into what's called the Krebs cycle. They enter what's called the Krebs cycle, which generates another 2 ATP. And then, and this is kind of the interesting point, there's another process that you can kind of say happens after the Krebs cycle, but we're in a cell and everything is bumping into everything all of the time. But it's normally kind of viewed to be after glycolysis in the Krebs cycle. And this requires, oxygen. So let me be clear. Glycolysis, this first step, no oxygen required, or doesn't need oxygen. Oxygen, it can occur with oxygen or without it. Oxygen not needed. Or you could say this is called an anaerobic process. This is the anaerobic part of the respiration. Let me write that down too. Anaerobic. Maybe I'll write it down here. Glycolysis, since it doesn't need oxygen, we can say it's anaerobic. You might be familiar with the idea of aerobic exercise. The whole idea of aerobic exercise is to make you breathe hard because you need a lot of oxygen to do aerobic exercises. So anaerobic means you don't need oxygen. Aerobic means it needs oxygen. Anaerobic means the opposite. You don't need oxygen. So glycolysis, anaerobic, and it produces 2 ATPs net. And then you go to the Krebs cycle. There's a little bit of setup involved here, and we'll do the detail of that in the future. But then you move over to the Krebs cycle, which is aerobic. It is aerobic. It requires oxygen to be around. And then this produces 2 ATPs. And then this is the part that, frankly, when I first learned it, confused me a lot. But I'll just write it in order the way it's traditionally wrote. Then you have something called, we're using the same colors too much, you have something called the electron transport chain. Electron transport chain. And this part gets credit for producing the bulk of the ATPs. 34 ATPs. And this is also aerobic. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can produce a little bit of energy, but if you don't, but it's nowhere near as much as you can produce once you have the oxygen. And actually, when you start running out of oxygen, this can't proceed forward. So what happens is some of these byproducts of glycolysis, instead of going into the Krebs cycle in the electron transport chain where they need oxygen, instead they go through a side process called fermentation. Fermentation. For some organisms, this process of fermentation takes your byproducts of glycolysis and literally produces alcohol. That's where alcohol comes from. That's called alcohol fermentation. And we, as human beings, I guess fortunately or unfortunately, our muscles do not directly produce alcohol. They produce lactic acid. So we do lactic acid fermentation. Let me write that down. Lactic acid. That's humans and probably other mammals. Humans. But other things like yeast will do. So yeast will do alcohol fermentation. Alcohol fermentation. So this is when you don't have oxygen. It's actually this lactic acid that if I were to, sprint really hard and not be able to get enough oxygen, that my muscles start to ache because this lactic acid starts to build up. But that's just a side thing. If we have oxygen, we can move to the Krebs cycle, get our 2 ATPs, and then go on to the electron transport chain and produce 34 ATPs, which is really the bulk of what happens in respiration. Now, I kind of said this as an aside that, to some degree this isn't fair, because while these guys are operating, they're also producing these other molecules, or they're not producing them entirely, but what they're doing is they're taking, and I know it gets complicated here, but I think over the course of the next few videos we'll get an intuition for it. In these two parts of the reaction, glycolysis and the Krebs cycle, we're constantly taking NAD, is NAD+, and we're adding hydrogens to it to form NADH. And this actually happens for 1 molecule of glucose, this happens to 10 NADs, or 10 NAD pluses to become NADHs. And those are actually what drive the electron transport chain. And I'll talk a lot more about it and kind of how that happens and why is energy being derived and how is this an oxidative reaction and all of that, and what's getting oxidized and what's being reduced. But I just wanted to give due credit. These guys aren't just producing 2 ATPs in each of these stages. They're also producing, actually combined, 10 NADHs, which are then used, which each produce 3 ATPs in an ideal situation, the electron transport chain. And they're also doing it to this other molecule, FAD, which is very similar, but they're producing FADH. Now, I know that all of this is very complicated. I'll make videos on this in the future. But the important thing to remember is cellular respiration, all it is taking glucose and kind of repackaging the energy in glucose and repackaging it in the form of, your textbooks will tell you, 38 ATPs. And if you're taking an exam, that's a good number to write. It tends to, in reality, be a smaller number. And it's also going to produce heat. Actually, most of it is going to be heat. But 38 ATPs, and it does it through 3 stages. The first stage is glycolysis, where you're just literally splitting the glucose into 2. You're generating some ATPs, but the more important thing is you're generating some NADHs that are going to be used later in the electron transport chain. Then those byproducts are split even more in the Krebs cycle, directly producing 2 ATPs, but that produces a lot more NADHs. And then all of those NADHs are used in the electron transport chain to produce the bulk of your energy currency, or your 34 ATPs.
So let's talk about the whole point of cellular respiration. The whole point is to take the things that we eat and break them down to make energy so that our cell can do work. And anytime we talk about cell energy, we're going to talk about ATP, which is adenosine triphosphate. Okay, so everything that you eat, your cell uses to have energy to perform work. In our cell parts lecture, we talked about the mitochondria, which was the power plant of the cell, so this should be at least somewhat familiar to you. These are these little kidney bean looking guys in the cell. And they're the site of aerobic cellular respiration, or the place where the energy is going to be made in the cell. All right, 2 types of respiration, and really they depend on whether there is enough oxygen available to the cell or not. Okay, so over here on the left, we're going to talk about aerobic respiration. If you're doing aerobic exercise, you're running, you're jumping, you're using a lot of oxygen, remember aerobic respiration also requires oxygen. So that's kind of the highlight. Aerobic requires oxygen. Aerobic steps are always going to happen in the mitochondria. It's going to make lots of ATP for the cells. So you'll see me refer to this as Plan A. It's the ideal plan. A couple steps that are aerobic are going to be conversion, citric acid or the Krebs cycle, and the electron transport chain, which we'll all talk about in a second. The next over here on the right is anaerobic, and anaerobic is kind of Plan B. This is when you don't have enough oxygen available. You can still make a little bit of ATP, but not a whole lot. This is going to occur in the cytoplasm, and our two steps are going to be glycolysis and fermentation. Along the way, we're going to pick up two chemicals, NADH and FADH2. Think about these as little shopping carts that we collect along the way, and during the steps, we're going to fill them with energy that when we get to the electron transport chain, we're going to convert that energy into directly ATP, which is our cell energy. All right, the 4 steps of respiration: glycolysis, conversion, citric acid, sometimes called the Krebs cycle, and then the electron transport chain. Cellular respiration is something that you need to look over a couple of times. You're not going to understand it the first time. You may not even understand it the second time, but hopefully after a couple times of hearing it, it will start to at least make sense to you. All right, aerobic cellular respiration, remember that's the one we have to have oxygen for. That's going to be our Plan A, our ideal way of making energy. All right, so our first step is glycolysis, and literally this means the breakdown of glucose. Well, what's glucose? That's sugar. It's in the food that you eat. Okay? So that glucose is going to be broken down into 2 pyruvate molecules. You're going to make 2 ATP, which is our cell energy, and then 2 NADH. Remember, that's one of our shopping carts. Because it's anaerobic, it happens in the cytoplasm of the cell. After glycolysis, we're faced with kind of a crossroads, an important decision. So that pyruvate that we just made has a decision to make here. If we have enough oxygen available to us, remember we want to go to our Plan A, which would be going on to conversion. If we don't, the only other option we have available to us is fermentation, which we'll talk about at the end, so just keep that in your head. All right, so our next step, which is conversion, that pyruvate that we just made is going to turn into 2-acetyl-CoA. And during this step, you're going to generate 2 more of those little shopping carts, the NADHs, and carbon dioxide, which remember is our gaseous waste product. It's an aerobic step, so remember it requires oxygen, and if it's aerobic, it has to be in the mitochondria. Next step is the citric acid or the Krebs cycle. And during this step, the acetyl-CoA that we just made are going to be broken down. If you do happen to take biochem after Bio 101, you'll have to learn all the fun steps that occur in here, but for our purposes, just know that acetyl-CoA is broken down. Once that's broken down, you're going to generate a whole lot of stuff here. 2 FADH2s, remember those are our shopping carts. 6 NADHs, more shopping carts. 2 ATP, so we're making some energy directly. And then carbon dioxide, that gaseous waste product that we exhale. It happens in the mitochondria, so it has to be aerobic. All right, now for our payoff stage. This is the last stage called the electron transport chain. Remember we said all the energy that we were storing in those shopping carts, we're going to kind of dump out on the cash out counter and we're going to turn it into ATP. So let's talk about where we collected those along the way. Okay, down here at the bottom. So we made 2 NADHs in glycolysis, 2 in conversion, and then 6 in the Krebs cycle. If you put those all together, that gives us 10 total NADHs that we made. The conversion rate for NADHs to ATP is that for every 1 NADH, we're going to make 3 ATP. Okay, so we have a total of 10. 10 times 3 gives us 30 ATP that we just made. One more shopping cart was the FADH2. The only time we picked that up was 2 in the Krebs cycle. The exchange rate for that is for every 2 FADH2s, we're going to make 4 ATP. And all we have is 2, so that would give us 4 ATP. All right, let's put that together. 30 plus 4 gives us 34 total ATP that we made in the electron transport chain. Compared to those little measly 2 here and there that we've been picking up, 34 is an awful lot of energy for our cells. Okay. So let's put that together for aerobic cellular respiration. Remember, we made 2 ATP directly in glycolysis, 2 directly in the Krebs cycle, and then we just made 34 in the electron transport chain. So if we put those all together, that gives us 38 total ATP. This is a magical number to remember because I promise you your instructor will ask you how many total ATP production in aerobic cellular respiration. All right, let's go back to that. Remember I told you to keep that fermentation in your head? This is the plan B part. Okay. That pyruvate that we made from glycolysis, remember it had a decision to make here? Well, before it had enough oxygen available, so it could go on to conversion. But let's say there's not enough oxygen available. The only option we have at this point is going on to fermentation. And remember, it's our backup plan, so we're not making nearly as much energy as we did in aerobic, but we're still making some. Okay, so in fermentation, remember, we do not need oxygen for fermentation to occur. There's 2 types. The first is alcohol fermentation, okay, and that's going to occur in yeast, and it will give us alcohol and carbon dioxide. The next is lactic acid, and this occurs in our muscle cells. Okay, the result is going to be lactic acid or muscle fatigue. And if you look there, we're only making 2 ATPs per glucose molecule. So that's not really a whole lot compared to that 38 that we just made. But again, it's a backup method. All right, let's review. All right, so we have a bunch of our steps here. Glycolysis. Remember, in glycolysis, glucose is broken down into pyruvate. All right, let's say we do not have oxygen available to us. What's the next step out of fermentation, conversion, Krebs cycle, and the electron transport chain are we going to go to? Hopefully you said fermentation. And what are our 2 types of fermentation? Good. Alcohol and lactic acid. Okay. All right, let's go back. Let's say we do have oxygen available to that little pyruvate. Where's he going to go next? Good, conversion. And in conversion, remember, we're converting that pyruvate into acetyl-CoA. All right? That acetyl-CoA is going to go next to the Krebs cycle. In the Krebs cycle, we're going to make a lot of FADH2s, NADHs, which are then going to take their energy to the last step, which would be the electron transport chain. And then they're going to exchange that energy for ATP, or cellular energy. Okay. This is a summary chart of everything that we just talked about. This would be a really good thing to memorize. It might help you learn to put things where they belong in the course of the aerobic respiration. All right, so we have our ATPs, our NADHs, and our FADH2s. And then we have our 4 steps here. So in glycolysis, we made 2 ATP, 2 NADHs. Conversion, we made 2 NADHs. Krebs cycle, remember we made a whole lot of stuff in that step. 2 ATPs, 6 NADHs, and 2 FADH2s. And then finally in the electron transport chain, we made 34. That was our big payoff stage of ATP. So 34 plus 2 plus 2 would give us a total of 38 ATP for aerobic cellular respiration. Again, I would encourage you to read this in your chapter, maybe watch this a few times. Don't get frustrated if you don't understand it at first. But I hope this helped.